everyone. In the previous session, we discussed about in vitro diagnostic devices part 1. In today's session, we are going to discuss about in vitro diagnostic devices part 2. In this, we are going to discuss about three major devices: ELISA machine, biochemistry analyzer and urine analyzer. To know more about ELISA, we must know what is immunoassay. Immunoassay is a test that uses antigen-antibody complex as a means for generating a measurable result. An antigen-antibody complex is also known as immune complex. An immunoassay test utilizes the immune complex when an antigen and antibody is brought together. One such technique is ELISA or enzyme-linked immunosorbent assay test. Now let's see the definition. ELISA or enzyme-linked immunosorbent assay is a plate-based assay technique designed to detect and quantify soluble substances like proteins, peptides, antibodies and hormones. This technique is also described with other names like enzyme immunoassay. Now let's see the working principle of ELISA. ELISA uses an enzyme to detect the presence of antigen-antibody binding. The enzyme converts the colorless substrate chromosome to a colored product indicating the presence of antibody-antigen binding. An ELISA test can detect the presence of either antigen or antibody based on how the test is being designed. ELISA was introduced in the year 1970 and it has become one of the rapidly accepted technology. Now let's see the technical flow of ELISA. This picture clearly depicts the technical flow of ELISA. In the cuvette, captured antibody are present. In the first step, antigen is added to the cuvette. Once the antigen gets bind to the labeled antibody or captured antibody, labeled antibody is being added to the cuvette. Once the labeled antibody is attached to the antigen, substrate is being added. The substrate then attaches or binds with the antibody. The signals from the attached substrate is being detected in order to monitor or indicate the presence of antigen-antibody binding in the given sample. There are two types of ELISA or enzyme-linked immunosorbent assay tests: qualitative and quantitative. Qualitative ELISA determines the antigen or antibody is present or absent. Quantitative determines the amount or quantity of the antibody present in the given sample. The highest dilution of the specimen, usually serum, which gives the highest positive reaction in the ELISA test. There are different applications of immunoassay. They are mainly helpful in the diagnosis of different diseases. They are also highly useful in detecting the severity of autoimmune diseases and immunodeficiency diseases. They are also helpful in detecting the hypersensitivity and tumor. The next device we are going to discuss is biochemistry analyzer. A biochemistry analyzer or a clinical chemistry analyzer is a device used to measure metabolites present in the blood. An automated biochemistry analyzer is a device used to measure clinical and other characteristics of a number of biological liquids or fluids with a quickly manner and with a minimal human assistance. These properties measured will be highly useful in the diagnosis of diseases. An autoanalyzer sequentially measures the blood chemistry through a series of steps like mixing, reagent reaction and colorimetric measurements, and finally displaying the measurements on a graphic recorder or a computer. Now we'll see the components of an autoanalyzer. The principal parts of an autoanalyzer are the following. They are simpler, proportionating pump, dialyzer, colorimeter, constant temperature bath and recorder. Now let's see the working of an autoanalyzer. The flow of the analytical stream is being directed through the plastic tubings from one module to another. The samples are now loaded into the cup of the sampler and the channels of the proportionating pump will be aspirating them as well as diluting them. The diluted serum is run through the dialyzer unit and the pump introduces suitable reagent through the other side of the dialyzer. The two streams run side by side with being separated only by the dialyzing membrane. A portion of dialyzable constituents of serum passes across the membrane to the reagent stream. Further treatment like incubation at constant temperature is given at the constant temperature bath. The intensity of the color developed is recorded by the colorimeter and is recorded in the recorder. Now we'll see the significance of autoanalyzer. Multiple instruments have been developed with multiple channels for around 18 constituents including non-dialyzable proteins, enzymes like alkaline phosphatase and electrolytes. Around 60 to 300 samples can be investigated per hour for around 18 parameters. Autoanalyzers have gained much popularity in the routine biochemical analysis because of their speed, flexibility of methodology, high or increased quantum in clinical biochemical analysis, easy operation and accuracy of results. The next device we are going to discuss is urine analyzer. Urine analysis is a valuable tool in diagnosing and monitoring renal as well as urinary tract illness. Urine analysis is typically a moderate to high sample volume test in general chemical laboratory with a representation of around 30% of the total samples received. Routine urine analysis consists of macroscopic examination, clinical analysis and microscopic urine sediment examination. A regular urine analysis consists of the following tests: color, odor, clarity, pH, specific gravity, glucose, proteins, leukocytes, bacteria, squamous cells, red or white blood cells and so on. Automatic urine analyzer. Several automated instruments are currently developed to standardize sample processing, biochemical test strip analysis, microscopy analysis and to report results. Automated urine cell analyzers aspirate, mix, dilute and stain urine to classify the urine sediment particles. Automated urine systems perform a complete urine analysis which consists of microscopic, physical and chemical parts of the routine. Benefits of urine analyzer. Urine analyzer consists of a whole host of chemical and microscopic tests and it is a useful tool in the diagnosis of diseases like urinary tract infections and renal infections and also other infections or diseases of the body that results in the formation of compounds in the urine that can be detected at the abnormal stages or levels. Now we have almost come to the end of the session. Hope you all understood about the concepts we have discussed about the three devices: ELISA machine, biochemistry analyzer and urine analyzer in this session. In the upcoming session, we are going to discuss about the summary of diagnostic devices. Thank you.